possibility of creating DNA, the blueprint of life, in a laboratory setting. DNA, or deoxyribonucleic acid, is more than just a string of letters. It's the master plan, the recipe book, the code that instructs every living creature, from the smallest bacterium to the largest whale, on how to live, grow, and function. But could we, as curious scientists and innovators, recreate such a complex molecule in our own laboratories? Could we take the building blocks of life itself, mix them together, and construct a strand of DNA from scratch? This is a question that has intrigued scientists for decades and continues to be a focal point in the field of synthetic biology. It's a question that requires us to delve deep into the world of molecules, genes, and life itself. Now let's delve into the complex structure of DNA to better understand the challenges of synthesizing it. At its core, DNA is composed of 4 nucleotide bases arranged in specific sequences. These bases, adenine, thymine, cytosine, and guanine, are the unsung heroes of life, carrying the codes that shape every living organism. Now, picture a ladder. The steps of this ladder represent these bases, which pair up in a very particular way. Adenine always pairs with thymine, and cytosine always pairs with guanine. When these base pairs come together, they form the rungs of our DNA ladder. But a DNA molecule isn't flat like a regular ladder. Instead, it twists around in a shape known as a double helix. The sides of this twisted ladder are made up of alternating sugar and phosphate molecules. These sugar-phosphate backbones hold the base pairs in place and give the DNA molecule its structural integrity. Think of the DNA structure as a language. The bases are the letters, the sequences of bases are the words, and the whole genome is like a book telling the story of an organism's life. The language of DNA is simple in its alphabet, but incredibly complex in its grammar and syntax. It's this complexity that makes DNA so powerful, but also so challenging to replicate. But DNA isn't static. It's a dynamic molecule that constantly unzips and rezips as it replicates itself. During this process, the base pairs split apart, each strand serving as a template for a new partner strand. This is how our cells make new DNA, accurately copying the base sequence so the new molecule is an exact replica of the original. Creating DNA in a lab isn't a simple task. It involves a multitude of challenges. And that's putting it mildly. DNA, as we've seen, is a complex structure, an intricate sequence of nucleotides wound into a double helix. To replicate this, we need more than just the chemical components. We need precision, accuracy, and a deep understanding of how these components interact. Consider the nucleotides, the building blocks of DNA. We have adenine, thymine, cytosine, and guanine. Now, imagine trying to assemble these in the exact sequence found in nature. It's like putting together the world's most complex jigsaw puzzle, where even a single misplaced piece can change the entire picture. And that's just the start. Once the sequence is assembled, we then have to form the double helix, a structure so intricate that it's been likened to a twisted ladder or a spiral staircase. Modern techniques such as solid-phase synthesis allow scientists to build DNA strand by strand. This process lets us control the sequence of nucleotides with utmost precision, but it's not as simple as it sounds. The process is complex, requires sophisticated equipment, and demands a deep understanding of molecular biology. And then there's the challenge of length. We can replicate short strands of DNA reasonably well, but as we try to create longer strands, the potential for errors increases. It's a bit like typing a long document without a spell check. The longer the document, the higher the chance of making a typo. And in DNA synthesis, even a single error can render the synthetic DNA useless. So the challenges are numerous and daunting, but they're not insurmountable. With each passing day, we're making strides in understanding the intricacies of DNA, in honing our techniques, and in improving our equipment. Yes, it's difficult. Yes, it's complex. But it's also fascinating, a testament to the wonders of life and the marvels of science. The accuracy of DNA replication is paramount. Even a small mistake can render the synthetic DNA useless. And yet we persist in our quest to understand and replicate the blueprint of life. Beyond the scientific challenges, creating DNA in a lab also raises important ethical questions. Synthetic biology and artificial DNA synthesis are not merely scientific endeavors. They also present us with a host of ethical considerations that we must grapple with as we advance in our understanding and capabilities. First off, let's consider the potential for creating customized organisms. This could range from bacteria engineered to produce beneficial compounds, to completely new species designed from scratch. While these could have tremendous benefits, such as helping us tackle disease or environmental issues, they also raise serious concerns. What are the ecological implications of introducing such organisms into the environment? Could they outcompete or harm existing species? And what about the potential for misuse in creating harmful organisms? Then there's the question of manipulating existing life forms. We're already seeing this with techniques like CRISPR, which allow us to edit the genes of living organisms. This has huge potential in medicine, for instance, in curing genetic diseases. But it also opens a Pandora's box of ethical dilemmas. Should we be altering the genetic makeup of an organism, potentially changing its very nature? And what about the possibility of creating designer babies, where parents could choose their child's traits? Such a scenario raises a myriad of ethical, social, and philosophical questions. And finally, there's the matter of ownership and control. If a scientist creates a piece of DNA or an entire organism in the lab, who owns it? Can it be patented? And who gets to decide how these creations are used or whether they're released into the environment? Navigating these ethical waters is no easy task. It requires ongoing dialogue among scientists, ethicists, policymakers, and the public to ensure that the pursuit of knowledge and innovation doesn't override important ethical considerations. Striking a balance between scientific exploration and ethical responsibility is crucial in this field. While DNA is fundamentally a chemical entity, its replication in a laboratory demands more than just a mixing of chemicals. It's an intricate dance of science and art, a delicate balance of precision and understanding. In our exploration today, we've delved into the structure of DNA, a double helix composed of 4 nucleotide bases: adenine, thymine, cytosine, and guanine, connected by phosphate-sugar bonds. This unique structure, akin to a twisted ladder, contains the blueprint for life, and recreating it is no small feat. We've learned that synthesizing DNA involves more than just combining these nucleotide bases. It requires a deep understanding of the molecule's composition and a meticulous control over the sequence in which these bases are arranged. Modern techniques, like solid-phase synthesis, have made it possible to build DNA strand by strand, but even with these advancements, the task is complex, requiring sophisticated equipment and expertise. Moreover, as we move towards the creation of longer DNA strands, the challenges multiply. The potential for errors increases, and even a minute mistake in the sequence can lead to functional changes or render the synthetic DNA useless. This underlines the importance of accuracy and precision in the replication process, highlighting the sheer complexity of DNA synthesis. But beyond the scientific challenges, we've also touched upon the ethical considerations that accompany the prospect of creating DNA in the lab. Synthetic biology and artificial DNA synthesis open up a world of possibilities that were once the stuff of science fiction, from creating customized organisms to manipulating existing life forms. However, these advancements also raise important ethical questions that we must grapple with as a society. In conclusion, the art and science of DNA creation is a complex and intricate process.